Today's lecture focuses on the organs of the immune system. The organs of the immune system are listed on this slide and divided into two broad categories, those referred to as the primary lymphoid organs and those referred to as the secondary lymphoid organs. The primary lymphoid organs listed in red play a central role overall in the immune response. They specifically are the sites of hematopoiesis and do allow for some maturation of lymphocytes to take place. The secondary lymphoid organs are sites where mature lymphocytes interact with antigen. And those are listed here in blue for you on the slide. In addition, with the secondary lymphoid organs, there are some different levels of organization associated with cells and cell types in the secondary lymphoid organ itself. Some of the secondary lymphoid organs have a lower level of organization of cells, basically just a collection of leukocytes in a target location. And we will look at some specific examples of that as we continue on with the lecture. Additionally, there are some secondary lymphoid organs that have a more organized structure of cells referred to as a lymphoid follicle. The lymphoid follicle comes in two forms, primary lymphoid follicle and secondary lymphoid follicle. Primary lymphoid follicles, again, have a higher level of organization of cells compared to the collection of cells I just mentioned. The primary lymphoid follicle typically contains dendritic cells, naive B cells, and again, naive B cells are going to be those that are not yet activated by antigen. A typical secondary lymphoid follicle is what forms or is produced after activation of the naive B cell. This is where B cell proliferation takes place. We also find some non-dividing B cells, helper T cells, and macrophages and dendritic cells in a secondary lymphoid follicle. So to summarize, there are two levels of organization of cells for secondary lymphoid organs. There are those that have a lower level of organization that essentially are just a collection of cells, versus something where there's a much higher level of organization referred to as a lymphoid follicle. So we'll focus on the primary lymphoid organs first. There are only two of them. The first one I want to discuss is the bone marrow. The bone marrow is a flexible tissue in the interior of a bone, and you can see that in the top portion of this particular slide in the panel associated with the long bone. In the center of the long bone, again, is where you would find the component that is playing a role in hematopoiesis. So the bone marrow, again, is specifically the site of hematopoiesis, and this is why this is referred to as a primary lymphoid organ, because this is an essential component for leukocyte development. In the bottom two figures of this slide, you can see some cells that are stained in order for identification of cell types to occur. In the left-hand figure, you can see some of the cell types that are identified, and again, for us, the layperson, it might be difficult to identify specific cell types. However, someone who is trained in this particular field could utilize the staining technique and identify different distinct leukocytes within the bone marrow. On the right-hand side is an unlabeled figure. However, this one has very clear differentiation in the staining pattern, which I thought was a really nice way to show the complexity of the different types of leukocytes that are found within the bone marrow. Within the bone marrow is where we find the hematopoietic stem cells, and again, essentially what is happening here is this is the site of hematopoiesis that will result in the production of the cell types that we discussed yesterday. The second of the primary lymphoid organs is the thymus. The thymus is a flat, bilobed organ that sits above the heart in a human vertebrate system. It is in fact divided into lobules that are separated by connective tissue known as trabeculae. Each of the lobes contains what's known as a cortex that has an outer, densely packed collection of immature T cells, known as thymocytes, and a medulla, which is the inner sparse component that is full with thymocytes. T cells arise in the bone marrow. It is the thymocytes arise in the bone marrow and then travel to the thymus where they continue their maturation and development. Hence, this is why the thymus itself is referred to as a primary lymphoid organ. So again, to summarize, the thymus is the site of T cell development and maturation. One additional point that I want to mention about the thymus is a process that we know occurs later on in life. The peak size of the thymus occurs right around puberty. And after that point, the thymus begins to slowly decrease in size at around the age of 65 years. This is referred to as atrophy. And at this particular point, we oftentimes see an increase in cancers, autoimmune disorders, and opportunistic infections in individuals greater than 65 years of age. And that's partially because of this thymic involution that is taking place, essentially the thymus shrinking down and not working at its optimum later on in life. Next, we'll transition into the secondary lymphoid organs, but before we do that, I want to discuss the lymphatic system overall. The lymphatic system is a series of vessels that are found throughout the body and essentially parallel and are distinct from our cardiovascular system. In the lymphatic system, we have the movement of a fluid that is referred to as lymph. So let's take a few steps back. When we talked about blood in our last lecture, we discussed the fact that the non-cellular fluid portion is referred to as plasma. Plasma can seep through capillaries into tissues, and at that particular point is referred to as interstitial fluid. The fluid must go back into circulation, most oftentimes returns via venules, and can actually enter in through the primary lymphatic vessel system. Once the fluid has entered into the lymphatic vessel system, it is referred to as lymph. And it is this fluid that then allows for the circulation of immune cells in order to play a huge role in the immune response. With the lymphatic system, again, it is a separate vessel system, separate from the cardiovascular system. And it does have a series of capillaries that are found where the movement can take place between blood vessel capillaries, the tissues, and then the movement back into the lymphatic system, like you can see on the right-hand side. Interestingly, with the lymphatic vessel system, there is no specific organ that allows for the movement of the lymph throughout the lymphatic system. Drawing parallels to our cardiovascular system, we have a nice, strong organ known as the heart, and the job of the heart is to pump the blood throughout the cardiovascular system. The lymphatic system does not have such a pump. There are a series of one-way valves that basically don't allow for the lymph to move in both directions. It allows for unidirectional movement of the lymph. And it is dependent upon both smooth muscle contraction and relaxation for movement of the lymph to take place. In addition, there is compression of skeletal muscle that is really important for the movement of lymph. This might be why a lot of times you hear that exercise is really good for your health as well as your immune system. And that's because that exercise that you're doing, that compression of skeletal muscle, is allowing for the movement of the lymph throughout the lymphatic vessel system. So the lymphatic vessel system essentially acts as the main highway for the movement of immune cells throughout the host system. Now we know that immune cells can actually travel throughout the blood vessel system as well, and that is definitely true. However, the lymphatic system is specifically involved in allowing for those immune cells to travel throughout the body to play a role in fighting off or eliciting an immune response. As I mentioned in the previous slide, there are a series of one-way valves that allow for unidirectional movement of the lymph throughout the lymphatic vessel system. Up at the top of the figure on the left-hand side, you can see the lymphatic capillaries, where the lymph will actually enter as interstitial tissue in the tissue space and then enter into the lymphatic vessel system, where it is then referred to as lymph. And then it travels through larger lymphatic vessels, passing through lymphatic or lymphoid follicles, and we'll get to lymphoid follicles in just a minute. Throughout the lymphatic vessel system, you will also find structures that are secondary lymphoid organs known as lymph nodes. These are sites where immune response is mounted to antigens within the lymph. Typically, lymph nodes are packed with lymphocytes, macrophages, and dendritic cells. Antigens can be brought into the lymph node, trapped by phagocytic cells, and allow for interaction between the phagocytes and the adaptive immune response. There is a very distinct anatomy associated with lymph nodes, which includes a cortex, a paracortex, and a medulla. However, in this particular case, I'm not going to focus so much on each one of those individual components of the structural features of the lymph nodes. Again, what I want to focus on is that this is where the site of activation of B cells and T cells can in fact take place. So the job of the lymph nodes mentioned on the last slide is to trap antigen at the local tissue level and begin to elicit an immune response. What I want to discuss now is another secondary lymphoid organ known as the spleen. We know that the job of the spleen is essentially to filter the blood, but can also play a role in trapping and responding to systemic infections. There are no lymphatic vessels that enter into the spleen. There is, however, just an artery that enters into the spleen. And in this way, the spleen allows for the filtration of the blood to take place. There are two separate components associated with the spleen. There's what's known as the red pulp. This is where we find macrophages, red blood cells, and some lymphocytes. This is specifically, though, where old or defective red blood cells are destroyed. The white pulp, however, is the portion that is really important for eliciting immune responses, and that is where T cells, B cells, and macrophages are found, and specifically B cell and T cell activation can take place within the white pulp. Again, the spleen is a filtration unit for the blood, so what this does is allow for detection of systemic infections that have entered into the bloodstream of the host. Both lymph nodes and the spleen have a higher level of organization that I mentioned previously, referring to both the primary follicle and the secondary follicle. These are a higher level of organization of immune cells playing a role in eliciting an immune response. There are, however, some secondary lymphoid organs that have a lower or much less level of organization and is basically a collection of cells. The one specific example I want to refer to is listed on this slide. The mucosa-associated lymphoid tissue, referred to as MALT, is a very generic term that is utilized to describe these aggregates of cells that are potentially playing a role in the immune response. We know that there are some specialized components of mucosa-associated lymphoid tissues, and this is going to be components where mucous membranes line organs, things like the digestive system, the respiratory tract, the urogenital tract, collectively are referred to as MALT, and then if we're referring to a specific component, might have a specialized name, like bronchus-associated lymphoid tissue or gut-associated lymphoid tissue. Overall, it is a collection of cells that is involved in detecting a potential infection overall. Additionally, things like the Peyer's patch, which is part of the small intestine, the tonsils, the appendix, and the adenoids all fall into this lower level of organization, a collection of immune cells. So let's look at these mucosa-associated lymphoid tissues in a little more detail. Within the lining of the epithelium of mucous membranes, there are cells that are epithelial cells, as well as cells referred to as M cells. These are specialized cells that allow for antigen transport from the internal component of the lining to a collection of lymphocytes that can interact with a potential antigen. So M cells are not phagocytes. They do not take in and destroy the potential pathogen. They do, however, move that potential pathogen across the membrane to the collection of leukocytes so that interaction can in fact take place. And so that is the job of the M cells overall. Now I want to quickly review things that we discussed in previous lectures, and that is the two main branches of the immune system or the immune response, known as the innate immune response and the adaptive immune response. Just hopefully you recall, the innate immune response is the first line of defense, includes non-specific mechanisms for identification of pathogens, typically referred to as patterns that can be identified. The main cell types involved in the innate immune response are phagocytes, as well as additional molecules that play a role in recognition. The innate immune response is present before an infection takes place. It's always present, ready and waiting to be able to detect a potential infection. The response time is typically immediate, can occur within minutes or hours, and there is no memory component associated with the innate immune response. With the adaptive immune response, we have the second line of defense. There are highly specific mechanisms involved, and there is the detection of very minor differences in molecular structures that allow for species-specific identification in the adaptive immune response. The cell types that are mainly involved in the adaptive immune response are lymphocytes. There are antigen-specific receptors involved and antibodies involved in the adaptive immune response. The adaptive immune response develops in response to an infection and is species-specific. Typically, the response time takes several days, and there is a persistent memory component. That is, once the adaptive immune response has been kicked in, you have memory cells that can detect that same pathogen upon a subsequent exposure. So now what we're going to do is do a quick overview of the innate immune response before we actually jump into the next lecture where we look at the innate immune response in a high level of detail. This slide and figure is meant to be an overview of the innate immune response. And from the first look of the slide, there's a lot going on. So hopefully what we'll be able to do is piece this apart and get a good handle on what's happening in the innate immune response. Recall the very first lecture I mentioned that there needs to be a recognition and a response. And that is clear in the innate immune system that recognition needs to occur in order for some response to be elicited. We can broadly talk about the functions associated with the innate immune response, and that includes recruitment of immune cells to the site of an infection. It also includes activation of the complement system, activation of the adaptive immune response, but more importantly, it is the identification and the removal of foreign substances that is the main function overall of the innate immune response. So let's go ahead and focus on one or the initial aspect of the innate immune response. One thing that must occur is that there has to be a distinction between self components and non-self components, something like a potential pathogen. How exactly does that happen? Well, there are receptors, molecular structures that are associated with many cell types, including leukocytes, that have what are known as pattern recognition receptors, or PRRs. So a PRR, or a pattern recognition receptor, does exactly what its name implies. It recognizes patterns associated with pathogens. These receptors detect very broad components, things like sugars, proteins, lipid-associated molecules that can be associated with particular pathogens. The recognition is patterns, broad structural motifs that are highly conserved and generally absent from the host. Now, with this recognition, with the receptors, what the receptors are binding to are known as pathogen-associated molecular patterns, or PAMPs, P-A-M-P-S. So PAMPs, pathogen-associated molecular patterns, are these structural components that are associated with pathogens that are typically distinct and not something found in the host. And it is these PAMPs that are recognized by receptors found associated with typical leukocytes. So what you see in this panel of the figure is that the PAMPs, the pattern-associated molecular patterns, are recognized by phagocytes. Neutrophils and macrophages typically are the number 1 and number 2 components that are recognizing these PAMPs. The PAMPs are recognized by pattern recognition receptors, and what this does is trigger or initiate the phagocytosis process. Additionally, a pathogen may undergo or encounter a different fate. If a phagocyte does not recognize and interact with the pathogen, there are other molecules that are produced by leukocytes that we mentioned in the previous lecture, known as antimicrobial peptides. Antimicrobial peptides are very short peptide molecules, meaning that they are a short stretch of amino acids, and there are a number of categories of antimicrobial peptides. Most of them, however, play a role somehow in damaging or disrupting the membrane of a pathogen. So a pathogen can encounter an antimicrobial peptide, which would then play a role in the destruction of that pathogen as well. Now, looking down the center of the slide, if a phagocyte does not interact with the PAMP via its pattern recognition receptors to initiate phagocytosis, or if an antimicrobial peptide doesn't play a role in the destruction of the pathogen, there are still additional components that come into play that can cause the overall destruction of the potential pathogen. This comes into play by molecules that are known as opsonins. Opsonins are molecules that tag or target an antigen for phagocytosis. So what you see in the center of the slide are 3 different examples of opsonins: mannose binding lectin, C-reactive protein, and complement proteins. They have the ability to interact with the pathogen and essentially coat the pathogen and help target it for phagocytosis. So the little spheres or dots that you see circling the pathogen represent these opsonins that are binding to the pathogen that help target it for phagocytosis. So what happens to a pathogen that has been opsonized? It promotes phagocytosis. Phagocytosis, again, will cause the destruction of that particular pathogen, but also we know that this is a link to the adaptive immune response. So the components that are broken down during phagocytosis are either released from the host system or are utilized as a link to initiate the adaptive immune response. And that's what you're seeing in the bottom panel of this particular figure. Hopefully what we've done is taken one small bite out of the entire process to give you a feel for what is happening with the innate immune response. And I suggest that you review this overview before we transition into the next lecture, where we look at the innate immune response in much more detail.